All right, so we have learned the process by which we make proteins. But one thing that we didn't discuss was that it takes a lot of energy to make all of these proteins. So in a eukaryotic organism like us, we don't want to waste energy making proteins that we don't need. In a prokaryotic organism, they also don't want to do that. But we have two different ways of regulating which genes get expressed, or in other words, which proteins get made and which proteins don't. And so gene expression is basically what we've been talking about, going from DNA to proteins, or from genotype or the genes to the phenotype, what it looks like. Gene regulation is the ability to turn on genes you need and off genes you don't. And this is part of the responding to stimuli that we talked about at the very beginning of the year with, what makes an organism alive. In prokaryotes, genes for related enzymes are controlled in units called operons. Okay, so there are two types of operons, and we will talk about those. You have a picture in your notes, so we'll go over that. Okay, but an operon is just a unit of genes that create enzymes which regulate the control of a DNA sequence. Okay, what I have here is an example of the lac operon, which is an example of an inducible. And basically what that means is when there is the presence of a certain thing, in this case it's lactose, this will turn on. When there's no lactose, it will turn off. Okay. So here is the operon. The operon goes from here all the way to here, and it contains a promoter, which we already know what that is. Okay. And this is on the DNA. Notice it contains something called an operator, and then the enzymes that you're trying to make, or the genes that try and make the protein. Now, if there is no lactose, okay, remember lactose is a sugar found in milk, okay? So let's say you haven't had any dairy, no milk, no cheese for like 24 hours, all right? We don't need to make the enzymes to break down that sugar anymore, because that's a waste of energy. So we are going to take something called a repressor, and we are going to attach it to an operator. So when the RNA polymerase comes over here to the promoter, because it's still going to attach to the promoter, it comes over to the promoter trying to make mRNA right here. If it comes over here to the promoter, it's going to stop. It's like a physical stop. Okay, the RNA polymerase can't go forward and will not make the enzymes. Okay, now, if we drink a big glass of milk and we've got a lot of that sugar is going to bind to this part of the repressor right here. And when it does, it's going to pull it off. Okay, so the repressor is pulled off of the operator, and then we can create those enzymes. Now, there is an opposite way that we can do this, which I don't have a picture of, called repressible. All right, I'm just going to write it over here, repressible. And this is the opposite of what we just did here, which is basically this is something that is on all the time. And when we don't need it, we can turn it off. So the example that you are going to see in your assignment is going to be of the tripoperon, and the tripoperon is tryptophan, which is an amino acid that's found in like turkey. And your body needs it every day, so we make it every single day. But if you eat it, like at Thanksgiving, you eat a big old bunch of turkey, we don't need to make our own tryptophan. So it turns it off, and it basically does it the opposite way. When we don't have tryptophan, the repressor's off. When we do, the repressor is on. All right, so now it's important for you to understand that every single cell in your body, except for your egg or sperm cells, have all of the genetic information that make you you. The DNA in every cell in your body is exactly the same. So how can we get a nerve cell? How can we have heart? How can we have different organs that do different things? That's a process called differentiation. So each cell has to have only certain parts of the DNA being shown or being expressed. The rest of it, like if you're a heart cell, you don't want the nerve cell proteins being made in your heart cell. Okay. First of all, it would mess up how the heart performs, but also it's a waste of energy. All right. So the differences between the different types of cell are due not to the DNA. The DNA in every cell is the same, but to selective gene expression. All right, so how do we do this? And this is just some examples. Remember, we talked about DNA being wound around histones and forming nucleosomes. When it is wrapped around histones, it is not being read or it is not being expressed. And there's a bunch of ways that we can actually keep the DNA wrapped around the histones. Methylation, acetylation, those are ways in which we can sort of turn off a gene. The prime example of this is X chromosome activation. So females have 2 X's and males only have 1 X. So in order to be a living person, you really only need 1 X, right? So in females, the other X actually gets turned off by packing up the other X in histones and adding some chemicals to it to turn it off. There's also some modifications that can happen as a result of epigenetic inheritance. And epi means above. So this is above the DNA. So this is things that don't really have much to do with the DNA that we're actually going to talk about in our genetics chapter. So we'll just put a pin in that for a second. And this is what that looks like. These are your histones right here. Then we've got them wrapped up in these nucleosomes right here. And then they're looped together. And so then we can turn off the whole chromosome if we keep everything wrapped up.
Remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
So in your notes, you have this checkpoint question, and I think we sort of answered that. So if you could just write that answer in your notes.
Most of the control of gene expression is achieved by regulating the frequency of transcription initiation. Various regulatory proteins bind to DNA and affect the ability of RNA polymerase to bind to the promoter site and carry out transcription. Efficient binding of the RNA polymerase increases the rate of transcription initiation. Gene expression can also be controlled by altering the splicing of exons in eukaryotes. For example, the same transcript made from one gene can be spliced differently to give rise to two very distinct protein products, calcitonin and calcitonin gene-related protein, CGRP. Gene expression can be regulated by controlling access to or efficiency of transport channels. The passage of an mRNA transcript across the nuclear membrane is an active process requiring that the transcript be recognized by receptors lining the interior of the pores. Many enzymes degrade mRNA. For example, specific nucleotide sequences near the 3' end may make mRNA targets for ribonucleases. Gene expression can be regulated by modulating the degree to which the transcript is protected or degraded. Many different proteins and amino acids take part in the translation process. The rate of gene expression can be regulated by speeding up or slowing down protein synthesis. The availability of any of the proteins or amino acids can alter the rate of the protein synthesis. Phosphorylation or other chemical modifications can alter the activity of a protein after it is produced. This is referred to as post-translational modification.
So this picture outlines everything we've been talking about as far as eukaryotic gene expression. So eukaryotic, that's really important. All right, so first of all, unpacking or not being able to unpack the DNA because of histones is a problem. When we do create, we transcribe, we cut up the RNA so we can make it, we can splice it any way we want. Remember, we add the cap and the tail, and as it said in the video, depending on the length of the cap and tail, could make it relatively unstable if you don't make it very long. So when it gets to the cytoplasm, it can be broken down. So mRNA just gets broken down into different pieces and gotten rid of if we don't need it anymore. So before you even transcribe it, we can do that. Before we translate it, once we make a polypeptide, we can modify it, right? Or we can break it down. We can cut it into pieces and get rid of it if we don't need it anymore. So these are all of the processes of regulating gene expression in a eukaryotic cell.